Welcome back, everyone. We have an update from Regen BioPharma Inc. It trades on the OTC Pink sheets under the symbol RGBP. It's a biotech company focused on developing innovative treatments using autologous cell therapies, RNA and DNA-based immunotherapy, and small molecules in the immune-oncology and autoimmune disease space. Happy to welcome back President CEO David Koos, Senior Scientific Consultant Harry Lander, and also welcoming Dinesh Singh. Nice to see you, gentlemen. Welcome back to the conference.
Good to see you. Thanks.
All right. I'll let you take it away, David.
All right. So this week, we're presenting primarily about Heme Accelerate, which is a therapy that we already have cleared an IND with the FDA. And we're looking at moving that forward into clinical trials. The reason behind that is that with the CAR T research we're doing, because that takes time between each step of the way, we also want to move forward on the Heme Accelerate, because it's something we have an IND for, and B, I think it fleshes out our portfolio a lot in terms of having a therapy that's actually ready for trial. So I think today we're going to, I'm going to have Harry ask Dinesh some questions about the Heme Accelerate. We're going to try and get to your questions today, but we are going to be backed up on time. So if for some reason we don't get to take questions, we will certainly do so at the next one when we have 30 minutes to cover the topics. And by that time, we'll also have an update on Duracar. So, all right, Harry, you got some questions for Dinesh.
Thanks, everybody. Yeah, let me introduce Dinesh. Dinesh, we've been looking for a world-class cell therapist and gene therapy expert, and we found one in Dinesh. I've known Dinesh for a while now, and he's a very accomplished cell therapy guy in the biotech space, in the academic space, so he knows the field very well. And because Duracar is taking, it takes time, and we have this clinic-ready drug, Heme Accelerate, we thought bringing Dinesh on board to help us move it forward would be a smart move. And so I would love for Dinesh to tell us a little bit about Heme Accelerate. Maybe, Dinesh, you could describe what kind of drug it is and what we plan on using it for in our first clinical trial.
Thank you, Harry, for taking me on board on Regen BioPharma. I'm really excited and looking forward to work on this wonderful drug. To me, it seems that, to be honest, I wonder why this drug hasn't reached the market yet. It's such a wonderful drug. Just for the sake of the people here on call, I'd like to describe it briefly. So, this drug is basically heterologous population of cells, mostly derived from the adipose tissue. And since it's a mixture of different types of cells derived from the same patient, mostly derived of immune cells, vascular epithelial cells, and so on and so forth, I think it has a huge potential moving into this space, not only for anaplastic anemia, which is our first indication that we have gotten an FDA approval, but also in the near future, expanding the portfolios in various other disease conditions.
So maybe you could describe, Dinesh, how it's prepared. Obviously, it's not a small molecule that you prepare in a factory and then you send it in a bottle. Maybe you could, because it's a cell therapy, perhaps you could briefly describe how it's prepared.
Yeah. So it's very simplistic in the terms that it's the patient's own cells that you take, and it is done by surgical removal of the fat tissue and mostly by lipoaspiration. And once you have this tissue, which is a huge mass of heterogeneous cells, you enzymatically digest it to remove the extracellular matrix, thereby purifying only the cellular population that you need. And to be honest, over the past 10 years, people have done huge amount of research using even modern techniques like single-cell genomics, single-cell profiling, you name it, they have done it. And to the core, that they have really identified more than 60 different types of cells, which was unheard of even when we started the program. Actually, I'm glad to know that Regen BioPharma had been a forward-thinking company, and they anticipated that the gold mine might be existing there. And here we are, that all the current scientific research published in high-profile papers like Nature, Cell, and Science are alluding to that fact that we're on the right track. So once you have the enzymatic digestion, you remove the debris, you collect the cells, and again, you can re-put the cells in the patient's, in our case, for the aplastic anemia. And you can imagine that initially the same cells might be there in the patient to start with. Now the question would be, why would it work? Is a fair question to ask. And in that sense, it is just like restarting a computer, that sometimes we have so many things going on, right? And the computer doesn't work, it hangs on every now and then. So only thing we have to do is just restart the computer. So isolating the cells from the same patient and purifying them, removing the debris and putting them back into the patient is just that's what we're doing, And based on the preliminary data that I have seen, it has huge, I think the results are very positive. And I'm very optimistic about this product.
Let me jump in real quick. Dinesh, beyond aplastic anemia, do you see other applications down the road that could be useful?
Yes. So actually, when I was reading all our hemaccelerate, right? I mean, chemotherapy, especially in cancer patients, it should be a huge boost, And there are some tweaking that we might have to do, but in cancer, just the simplest thing, without even any tweaking, the cancer-induced neutropenia is this condition where we can use it. Not only that, what I see is that if we are able to isolate the cells prior to chemo or radiotherapy, right, in the patient, and then that kind it can act as a, patient-derived, anti-cancer cells, because this tissue or hemaccelerate is a good population of cells, not to mention, people have anticipated to the extent of, say, 20 or 30% of even natural killer cells, which are, a good arsenal for cancer. So I see huge potential for this molecule moving forward.
Excellent. Anna, I'm curious. What do we have left? I'm trying to decide if I want to throw in another question for Dinesh, or we're going to probably carry it over to next month when we have a longer time period.
Yeah, feel free. Take a few more minutes, and then we can go with some viewer questions. So go for it. Okay,
cool. Harry?
Well, we were talking about a phase 1 clinical trial, and as we know, and we've talked about, we have an IND cleared by the FDA to begin when we're ready. And so, Dinesh, maybe you could talk a little bit about how long that would take, the phase 1 trial, and sort of maybe the steps that we would need to do to sort of get ready to enter patients into the first phase 1.
Yes. So approximately, again, it depends on the funding, but once funding is in, for the first phase, it will take around a year for the phase 1. And thereby, we would be moving into phase 2, which is a recruitment of more patients, activating 10 different sites across the US so that we have the requisite number of patients moving in our clinical trial, right? And that can take anything around 30 to 2 months, for the full study to be complete.
Okay, very helpful. And getting back to Heme Accelerate, so the bone marrow in aplastic anemia is not producing enough red blood cells, right? And so, in your view, what cells are in Heme Accelerate, whether they're stem cells, hematopoietic stem cells, endothelial cells, progenitors? what cells do you think that are in Hemaccelerate that can lead to curing aplastic anemia?
So that's a wonderful question, and also, to be honest, there are a lot of cells which assist, So it's not only that when you take a hematopoietic stem cells, it produces all sorts of cells, right? But it's not that you need all types of cells. You need a certain types of cells. So these taking the Hemaccelerate drug product, which has already a population of lot of immune cells, which otherwise the hematopoietic stem cells does not need to make, right? But whatever is there, right, your body cells, you need to facilitate them making the required type of cells, So in that context, the presence of, growth, like mesenchymal cells, so stem cells, which have some potency to make new types of cells, and also presence of other hematopoietic, regulatory T cells and so on and so forth, even NK cells, right? They provide a nice niche where the body cells are facilitated, and not over-jammed with producing everything, but only what the body needs at that time. So there is a clear distinction between that, And especially in aplastic anemia, where we are thinking of recruiting patients who otherwise does not respond to hematopoietic stem cells, this should be a boon.
Thank you. Thank you, Dinesh. David, do you have any other questions for now?
Well, I do, but unfortunately, I'm really trying to get to some of the audience's questions, which is backing us up a little bit. So I guess the Hemaccelerate, we plan on discussing that more in depth next month. And also, we should have an update on the DuraCar program for next month. So with that said, Anna, do you have questions for us?
Yeah, let's see if we can get a few questions in the next minute or so. Kristen Sokolowski asks if you're looking for people to participate in the study, or do you already have a pool of people?
I think that typically that's going to be the contract research organization's job, assembling the pool of people that are going to be used in the study.
Okay. We have a question from Greg. You previously discussed the potential of grants. Can you talk a little bit about when you might be applying for a grant, how many, and for how much?
Well, I think the grant idea is that there are definitely quite a few grants out there that we would be interested in. Some that we've looked at, it looks like their windows are somewhere around September. So unfortunately, the way the grants work is they're not just sitting out there on the shelf waiting for somebody to come along and say, here, we'd like to apply for this. They have certain windows of opportunity. So yes, the grant idea is still very important to us. I think that's really good. I think it's the idea is that it's non-dilutive, which is beneficial to shareholders, and including myself as a shareholder. If you have grant money, then those are shares that you didn't have to issue to finance the company.
And I know you mentioned you'll be speaking about this more in the next conference, but Marcy Gilbert wants to know what the results of the last DuraCar NR2F6 study was, if you can speak about those.
Well, okay, so the DuraCar, I'm stepping all over Harry's foot. So they're in the process of, I think the cells were being grown for the second round of studies there. And we expect to have those anytime, but we didn't have them in time for this conference. So we should definitely have them by the next conference.
All right. Well, thank you, guys. Do you have any closing remarks for our viewers watching you guys today?
Well, I'm very excited that we're finally able to move forward with a drug that's actually ready for clinical trials. That changes our game plan a great deal, because now we go to a clinical biotech company and not just a preclinical. I'm excited to be able to share as much as I can. I hope that it's not too disturbing because we ran out of time. We could keep going on this thing probably for an hour. But then, anyway, that's where it sits.
Well, great. I'm glad we'll see you back on in May with a longer presentation.
Yep. Thank you very much, Anna.
All right. Thank you guys for the updates. Okay, everyone, stay with us. Carbon Revolution had to push to May, but we'll be back with I2 Biopharma. We'll see you soon.